Hello, everyone. How's the volume? Can you hear me? Awesome. Well, welcome. Thank you for joining us tonight. My name is Montana. I work at the River Center as the education manager, so I get to put on fun programs like this. We're super excited to have you. I like to start our presentations with a little informal survey of our audience. So raise your hand if you've attended a River Center program before. Okay, raise your hand if this is the first one you've come to. Nice. We got some new faces in the audience. That's great. What about River Center members? Who here is a River Center member? Okay, raise your hand if you became a member today. Let's go ahead. Let's give a little clap. Woo! If you too want to get a little clap for yourself, we can sign you up right at the front desk. It's $35 for the year, covers your whole family, and it gives you discounts on programs. Sometimes that discount might be $2. Sometimes it might be, I think, up to $50. So it's definitely worth checking out if you want to come to some things on a pretty regular basis. Speaking of which, our next program you might be interested in is Tracking Notes, The Secret World of Mountain Lions. It's part of our film series. It'll be a documentary that we'll have here at the River Center October 16th. So that's a Thursday. I think it's 6 p.m. And again, that's $10 for members, $12 for non-members. So something to keep in mind. You can find out all about our programs on our website or receive our monthly newsletter and see what's happening here. Now, I'm not going to talk too much because I have some lovely folks who are going to do an introduction, but we're really happy to have Angelica and David and all the folks with the Fungi Festival here tonight. This is a fun partnership, new opportunity. They're dancing in the back. You can't see them, but they're going like this. Super excited. David Rogers is going to come on up and introduce Angelica and the festival. And he is the founder of the Olympic Peninsula Fungi Festival and Maddie's Mushrooms. This is their 4th year for the festival, which is exciting. It's pretty new, but it's already grown into this huge thing. And I love that we have it right here on the Olympic Peninsula. So let's give a warm welcome to David.
Good evening, everyone, and thank you for coming out. As Montana mentioned, my name is David Rogers. I'm the director and founder of the Olympic Peninsula Fungi Festival and the owner of Maddie's Mushrooms, a small gourmet mushroom farm here on the peninsula. I'd like to start by sharing a bit about the Olympic Peninsula Fungi Fest. Many of you might already follow us, but for those of you who do not, the event is coming up October 17th through 19th. We have some pre-fest forays and our Friday keynote dinner happening on Friday, October 17th. And then we'll be at the fairgrounds on the 18th and 19th with free access spaces, our education barn, vendor hall, food court, as well as ticketed presentations and workshops. Our Friday keynote dinner this year is featuring speaker Langdon Cook, and it will be held at the Field Arts and Events Hall downtown on the waterfront in Port Angeles. At the heart of the work we do at Olympic Peninsula Fungi Festival is highlighting fungi, an often overlooked and underappreciated kingdom of life that is absolutely crucial to our existence. From ecosystems and food systems to medicine and culture, fungi connect and sustain so much of what we do. And that's why events like this are so important. I'd also like to take a moment to thank Dungeness River Nature Center for hosting today's presentation and for highlighting nature and including fungi in that. I now have the pleasure of introducing today's speaker. One of the things we try to do at our festival and across the field of mycology is to make sure that those doing great and passionate work are given a platform. Dr. Amy Honan, one of our festival's directors of research, personally recommended today's speaker along with many other very loved mycologists. Angelica Bayless is an emerging mycologist whose journey began with mushroom cultivation and foraging in Colorado, and that has since led them to pursue a degree in mycology at the Evergreen State College. Angelica has already begun meaningful research going through and organizing many documented finds from the Evergreen campus um, and continues to share their love for fungi with the wider community. So please join me in welcoming today's speaker, Angelica Bayless.
Hello. Sound good? Everyone can hear me? Okay, perfect. So, as David said, my name is Angelica. I am currently going into my third year at the Evergreen State College, with my degree pursuit being mycology, as he mentioned. So, let's see. I don't know why it disappeared, but today I want to talk about fungal diversity, conservation, and community science. But first, I have a question to pose for all of you here today. This question is, why are you here? What brought you here? Now, feel free to sit with this for however long you need to. Personally, I am here because I truly, truly love fungi in all of their amazing forms. I'm here because, like David, some amazing people have given me this opportunity. And lastly, I am here to hopefully teach you all a little bit more about fungi and their amazing roles within all of our lives. Okay, there we go. So just a brief presentation overview. These are the topics I'll be covering. Now, the order of them has been changed a little bit, but still, this is what we will be going over. So firstly, I want to talk about what exactly makes a fungus a fungus. So firstly, they are eukaryotic organisms, and one of the most important aspects of fungi is that they are heterotrophic, which means that similar to us, they cannot produce their own food, so they need to outsource it. Meaning, the bottom word underneath heterotrophic, they are osmotrophic, which means that they go through several different processes to externally absorb their food. So they're breaking things down outside of their body before they're absorbing it. Another important thing about them is that they have chitinous cell walls, which chitin is the same material that the exoskeletons of crustaceans and seafood, stuff like that, and insects. That's what makes up their exoskeleton. Fungi grow from the filamentous tips of little threads that are called hypha. And when all of these threads get together, it's known as mycelium. So an individual thread, like a thread from a shirt, hypha. The shirt, mycelium. And then regarding mushrooms themselves, the example I always really like to go with is to compare them to apples. Now, you can think of the mushroom, like a portobello mushroom from the store, as being the apple from the apple tree. Sure, you know, it's what people see most often, but it's just a small part of the overall organism itself. And so the bulk of the organism is going to be the mycelium underneath, as you can see in the bottom left picture, similar to how the bulk of the apple tree is going to be the tree itself. Next slide. So now I want to talk about the big picture of fungal diversity. How many are there? Now, our first conservative estimate is based off of comparing plant and fungi ratios in very well-studied habitats. So these are pretty niche habitats where you can really easily examine everything that's there. And then our second estimate incorporates data from additional biodiversity hotspots, new habitats, and also changes to how we actually classify a species. And then on the less conservative side, as you can see, we have quite a range of what people think exist. So that first estimate, and really the second as well, 3.5 to 6.3 million species, is based off of DNA analysis primarily. So this is going to be people looking in soil samples, understudied habitats, stuff like that, to see what exactly is there through DNA analysis. So we're not necessarily seeing physical fruit bodies in all of these cases. It's just going to be DNA. And then that last bullet point, as you can see, is by far the most extreme. And so that is based off of including cryptic species, which a cryptic species is defined as being a species that is only recognized by DNA analysis. So regardless of how much you try, you can't separate what it is based off of what it looks like. You can only separate it by the DNA. And so now to kind of put that into perspective, comparing how much we actually do know, how many we have identified, that's going to be 155,869 individual unique species. So going back to the previous slide, we really don't know that much. Where am I in my notes? Okay, so within the overall fungal kingdom, there are 12 different phyla, or 12 different groups of classification. And there are two main phyla that make up virtually everything that we have identified so far. And that's going to be the Ascomycota and the Basidiomycota. And so both of those combined make up 97% of everything that's been identified thus far. And our image on the right kind of shows the different phyla. And you can see Ascomycota dominates the majority, with Basidiomycota being a good third. And then all of those small little colors up at the top are the other 10 phyla, making up only 3,300 species. To once again put that into perspective, across the board with this graph, I kind of separated it by the different estimates that we have. And so if we're going with our smallest estimate being 1.5 million species, we have only actually identified a little over 10% of that. And on the far side, with 13.2 million species, even less at 1.18%. So it can easily be assumed that there is a lot that we can learn, a lot that we don't know about. And so I have another question to pose. What could some of these identified fungi look like? I'm going to get a sip of water really quick as well. Just a brief pause. Okay. So firstly, we have mushrooms. Now, when I say the word mushroom, I usually assume that most people think of a tiny agaricus portobello mushroom from the grocery store. And what I really want to stress to you all today is that the world of mushrooms, actual physical fruiting bodies, is so much more diverse than just a small portobello mushroom. And so, as we know, mushrooms themselves are the reproductive structure of fungi. They hold the spores that start new colonies. And fungi that create fruiting bodies are primarily only found in the Basidio and Ascomycota. And so the two largest groups that we've identified, we've identified them because they produce fruiting bodies. So it's easier to see them. And then most fungi in and of themselves don't actually produce fruiting bodies. And I think this connects to how we really only see them within the Basidio and Ascomycota, because those ones are producing fruiting bodies. We find them more often. We're able to associate with them more. And everything else kind of slips under the door. We don't really see too many of them. Now, overall, I managed to find one estimate that said that something around 10% of all fungi that we've identified, and all fungi in general, produce fruiting bodies. However, I am taking it with a large grain of salt because I couldn't find the sources. And so I don't really trust it too much. So we're not really going to base too many things off of that. But I just wanted to mention that there's one estimate out there. And so another group to look at are lichenized fungi, also known as simply lichens. And lichenized fungi, in my mind, are old reliable. Regardless of weather conditions, regardless of how much moisture we have, I know that I can go outside and I can find a lichen on a rock. And so lichens in and of themselves are symbiotic organisms, and it is a relationship between two partners, two or more, really. And our partner number one is going to be a photobiont, which is a photosynthesizing partner. So that could be either an algae or a cyanobacteria. And then our second partner or third partner is going to be our mycobiont, which is going to be a fungus of some sort. And now there's a wide estimate between how many there are of lichenized fungi, and it ranges between somewhere around 10,000 to 18,000 different species. But it's also estimated that 17 to 30% of all fungal species could become lichens. Don't ask me how they do that. I don't really know, but that's another estimate that's been established. The next group of fungi that we're going to look at today are, in my mind, a little bit underappreciated. So both rust fungi and smut fungi are examples of plant parasites. And honestly, I just don't really see too many people talking about them, which is why I want to bring them up today. They're rather large, influential groups when it comes to agriculture, like growing food itself. And there's a lot of overlap between rust and smut fungi. So they both operate as obligate parasites of plants, which means that they require a host plant to both survive and to complete their life cycle. They both belong to the Basidiomycota phyla and pose rather large threats to agriculture. In the case of rust fungi, there are approximately 17,000 species estimated, and they're named rust fungi because they leave a brown, rusty appearance on infected plants. And a good way to tell if you have a rust or not is to just go up to the plant, try to rub it off, because the spores of rust fungi exist on top of the plant. And so if it's a rust, it'll rub off. If it's not, it'll stay. And then for smut fungi, there are around 5,000 species currently described. And it's also thought that at least 10% of the entire Basidiomycota phyla are made up of smuts, which is roughly the 5,000 species based off of what we've identified currently. And the name smut is given to this group of fungi because they create a black, sooty appearance on infected plants. And so smut, like kind of charcoal. And they're very well known to infect staple crops like wheat, rice, maize, sugarcane, where when they do infect these crops, they really are responsible for decreased crop yield, quality, and economic value in all of those different fields. I'm gonna get more water. Okay. The next groups are a little bit different than the previous groups I've been talking about, because in most cases, they don't necessarily produce a visible structure. So all of these groups could kind of be what I was talking about earlier, fungal species only identified through DNA analysis. But regardless of that fact, I think it's still important to cover them. So firstly, we have mold. Now, I am 100% sure that everyone in the audience has come into contact with mold. And it's, again, there's a large difference between how many species are estimated to exist, with a conservative estimate hovering around 20,000 species to 100,000 species. And I couldn't quite find a direct number of how many have been correctly identified. So these estimates of how many species there are is about as good as we're going to get right now. And they primarily belong to the Ascomycota, but they can belong to other phyla as well. There's no specific group that only creates molds. And then mildews are similar to mold, except a little different, as all things are. There's around 900 species of mildew, and they're primarily split between two different types. So we have powdery mildew, which starts out gray or white and then turns to a yellowish brown to black as it ages. And it's mostly found on flowering plants. And then downy mildew, on the other hand, starts out as a yellow spot and it slowly turns brown over time. Downy mildew is also primarily found on things like potatoes and tomatoes, among other common food crops. And so that's one that we don't really like as much as people who like to utilize those foods. And then yeasts are single-celled organisms, which means that every single life function that needs to happen for them can happen in that one single cell. So they don't need any specialized cells or anything like that like we do, just one. And then there are over 2,000 species of recognized yeasts, but between all of us, I think there's a lot more. And yeast is also very important to us as people because without it, we'd have no bread, we'd have no wine, we'd have no cheese. And it's responsible for like so many things that we as people enjoy. And so we've been talking a lot about big picture ideas of different types of fungi, different groups. So now I want to zoom in to our state here. So Washington State in general, as David mentioned, I'm from Colorado. So Washington State has just been incredibly diverse when it comes to fungi, which is such a nice change because, well, I like diversity. And so for reference, within Washington State, there are 3,451 vascular plants. So a vascular plant is going to be most all of what you see when you walk outside. Trees, shrubs, various things like that. And then combining our two numbers right here, the 2,537 species of macrofungi and the 1,195 species of lichenized fungi, we get 3,732 total fungal species found within Washington State. What does that mean? We have more fungi here than vascular plants. And just for a little bit of reference, a non-vascular plant is going to be something like algae, mosses, or liverworts, for example. But virtually all other plants that you might come across are vascular. So let's see. So now I want to talk about some of the things that fungi do for us and how we've used fungi as people. But first, I need to find my page of notes. I'm so sorry. Okay. So on the left side, we have a column that I like to refer to as creature comforts. Again, virtually all of these things, if they disappeared from our life tomorrow, we would be sad, but we would survive. Now, on the right side, however, that small column up in the top right, I like to refer to that as life or death. So firstly, we have penicillin, which has been a life-saving antibiotic for so many people. It was discovered in 1928, and so we've been using it since then. And then next we have statins, which are a group of cholesterol-lowering drugs. And since 1994, when these drugs became widely available, they have lowered cardiovascular disease by 25% within the US alone. And so think about how many people have needed these. It's a lot. And then our last one up there, cyclosporin, anyone who has ever gotten some sort of organ transplant and has had it happen successfully is usually able to thank cyclosporin for that because it is virtually an immune system suppressant. And so when you're getting this new organ into your body, it's going to prevent your body from attacking it. And so it really helps to prevent transplant rejection for anyone who's ever had a transplant. And all three of those life-saving drugs all originated from fungi. among many, many others, but I just wanted to highlight those three in particular. So next we have mycorrhizal fungi. Now, mycorrhizal fungi have a mutualistic symbiosis with plants. There are a few different kinds, but across the board, they help plants to uptake needed nutrients such as phosphorus and nitrogen. And in exchange, the fungi get things like sugars that the plant makes during photosynthesis. So they essentially hook onto the roots of plants, as you can see in that top right photo, and they extend the reach. And so anytime that there's water stress happening, anything like that, the fungi is able to go further than the plant's roots. And so it's able to bring water from farther away to help the plant survive. There are over 6,000 species of mycorrhizal fungi, and over 90% of land plants have and often rely on them to survive. And I quickly want to highlight the word mycorrhizae itself because I really like the translation. I'm kind of into translating fungal Latin names. I think it's really fun. It's a pastime of mine. And so myco, our first half of the word, translates to fungus. And then rhizae means root. And so the word mycorrhizae is literally meaning fungus root, which I think personally really accurately describes what they are. And then here are some local mycorrhizal fungi. Many people take interest in these species because, you know, some are culinary reasons, some are for beauty, some are for science. So in the top left, we have some beautiful chanterelles. Bottom left, we have Amanita muscaria. In the middle, we have tasty matsutake. Top right is going to be king boletes. And then the bottom right is going to be a truffle. Believe it or not, we have truffles here in the Pacific Northwest. I haven't found any. It's on my bucket list. And then the next partnership that I want to talk about are endophytes. Now, would you believe me if I told you that every single plant species that has ever been officially studied has been found to have at least one endophyte in them? Maybe, maybe not. So in particular, we're talking about fungal endophytes today. However, an endophyte is defined as being any sort of microorganism that lives asymptomatically within plant tissue. And so that could be bacteria, archaea, algae, maybe, similar things like that. And so again, to date, every single plant that's been studied, which at this point, I believe is virtually every plant on the planet, except for maybe some found in very niche habitats, has been found to have at least one or more endophytes. And they can be found within the plant organs, pretty much all the plant organs. So the roots, stems, seeds, and leaves. And then the interactions between plants and endophytes vary widely and are very diverse overall. But ultimately, the most beneficial thing that the plant or that the endophyte gets out of the relationship is protection. So the plant is essentially creating a home for the endophyte to live in where, you know, it might not otherwise have that home. And oftentimes for endophytes, they need to exist in some sort of plant to survive at all. They can't live outside of it. Now on the bottom, at first, it really seems like the plant gets the better end of the deal. And for the most part, it does. There are cases where endophytes act more as pathogens to the plant versus being beneficial. But overall, they're usually positive for them. And so some of the benefits that the plant gets is having the endophyte create useful metabolites and phytohormones. Where am I in my notes? So some of those useful metabolites are going to be something like phosphorus, iron, and nitrogen. And then the phytohormones help the plant to respond to environmental stressors. And they often help the plant to uptake more nutrients or to uptake them more effectively. And then there's also giving them higher resistance to pathogens, and then targeted pathogen and pest control through creating various antibiotics and metabolites that are able to increase the plant's defense system. And overall, I think that endophytes are super, super fascinating. I had maybe one week, one lecture my last school year that we covered endophytes, but I don't hear anything else about them any other time. So I kind of want to change that. I'm interested in them. So next, I want to take a moment to talk about fungal lifestyles. And I've begun to use the term lifestyles when describing this because, well, that's the term my mycology professors use, and I think it rather fits pretty well. And so a fungal lifestyle refers to the way a fungus interacts with its environment and how it gathers resources and nutrients. So firstly, we have saprotrophs. Now, a saprotroph can break down and consume dead or decaying matter. And these are commonly seen in the woods, usually growing from leaf litter or the duff, and often from dead, decaying logs. And so some examples of saprotrophs are going to be button mushrooms, shiitake, oysters, and turkey tail. And then next, we have the necrotrophs. Now, a necrotrophic fungus will infect a living organism, and then it'll slowly kill it off. And as it's doing that, it will consume the dead cells. So it's working similar in a way to a saprotroph, but it's doing damage to a living thing first. It's not waiting until it's dead. And then the next lifestyle we have is biotrophs. Now, biotrophs are virtually the opposite of necrotrophs, where they feed on the living host cells of the plant. And most of the time, if at all, it takes a really long time for them to kill off the plant. And so it's a bit different than the necrotrophs in that case. And then hemibiotrophs are kind of a combination lifestyle. So they have both a necrotrophic and a biotrophic phase. So they first start out operating as a biotroph, existing within the plant and feeding on living cells. And it then moves into a necrotrophic stage where it begins to kill off some of the cells and then still consumes those dead cells. So it doesn't need living or dead cells. It'll do both jobs. It'll always find a way to get what it wants, really. And so we've been talking a lot about various different types of fungi. I now want to shift the conversation towards a direction where we're discussing how we can increase our knowledge of biodiversity. And so this is going to be increasing the species that we've identified, increasing what we know, increasing awareness, virtually all that kind of stuff. And to do this, I have another question to pose. The question is, how can we utilize community science as a tool to document biodiversity? Now, can I get a raise of hands for who is familiar with community science? Oh, beautiful, great. So let me go to the next slide. So for those who are unaware, in a nutshell, community science, also commonly known as citizen science, refers to getting any community member involved in some sort of scientific action of any sort, really. Now, the bubbles on this screen, in my opinion, highlight the most important aspects of community science. So it's going to look like getting people engaged with nature and their local environments, fostering curiosity, learning, and stewardship. And it also, this is one of the things that I think is most important. It redefines who is able and allowed to participate in science. Oftentimes, there are going to be various barriers: education, where you live, age, all these various things that are going to be stopping people from incorporating science into their lives. But through community science, I think that it really helps to redefine that narrative. And it's my personal belief that community science is one of the most important tools for documenting and monitoring fungal biodiversity, and that anyone and everyone can be a community scientist. And one of the most popular and powerful tools for community science in this day and age is the app/website iNaturalist. Now, can I get a raise of hands for who is familiar with iNaturalist? Oh, great, beautiful. So for those who aren't familiar, iNaturalist is a community science platform where anyone can make and document observations of their local flora, fauna, and fungi. It was originally created in 2008, and it has since become a nonprofit in 2023. And their mission overall is to connect people to nature and to advance biodiversity science and conservation. They encourage this through action. So these actions look like getting out into nature and observing what's around you. And then you have the opportunity to record those observations via photographs and notes. And then it also allows you to pretty much describe what you're seeing in your understanding, regardless of what level that may be. And then the last part is by uploading this. And so you're able to upload what you're observing, and it allows for anyone, anywhere to really look at this observation and to see what you saw. So it's essentially giving a pinpoint that's saying, hey, look at me, look at me. I saw this thing here. And regardless of who's in this room, you can go and see what I saw there. Okay. So this map looks a little bit daunting at first. It's the main interface that iNaturalist operates on. So when you're typing, when you're making your login, when you're going in, this is what will pop up immediately. And it's essentially showing everything that's ever been observed with it since it was created in 2008. And so as we can see, to date, or rather last Saturday when I took this screenshot, there have been over 276 million different observations of flora, fauna, and fungi. Now, to make that even more specific, I have this screenshot. Now, with the map feature, it gives you a lot of different options for how to filter these observations. And so the previous slide was every observation ever. This slide is only the fungi observations. And so to date, also last Saturday, we have identified almost 17 million different fungal species, well, fungal observations, spanning almost 26,000 different species. So just in and of itself, iNaturalist is one of the most well-known and widely used tools for documenting species. And this kind of proves that. So now, to get even more specific, I was messing around with it and I wanted to see how many observations have been made in Squim itself. And so with this map, I just made a barrier around town, not really caring about how large it was. And so within that little barrier, there have been 384 fungal observations. And I'm sure if I did any species, any sort of creature, there would be a lot more observations. But these are just the fungi. And so there have been almost 150 different species seen just in this area. So next, I want to show you an example of an iNaturalist post. So this is one of my observations. I'm pretty sure this one was for a class project last year, if I'm recalling correctly. And so in each post, you are able to upload both photos and notes. And so the notes section is just some various good practice things to include in the notes that I'll get to a little bit later. And then photos are just going to be photos that I take of it. And so quickly, I want to get into the community taxon. So it's in that far right middle section. And so the community taxon is essentially what other iNaturalist members have agreed on regarding the identification of this species itself. And so that essentially means that other people went to this post, they saw it, and they were like, yes, I agree with you. That's what that is. And so having them do that is what makes this post research grade as well, which is right up here in this top middle section. Now, a research grade post means that, again, someone came across this post, they saw that I thought it was the pleated Marasmius, and they agreed 100%, no other opinions. Yes, you are correct. However, oftentimes it takes a little bit more for something to be research grade. So in general, the journey to becoming research grade can look like a lot of different things. And so if someone, instead of agreeing with me, disagreed with me, I was wrong, they're right. And let's say that they suggested a different identification, a different species, then it's essentially giving this observation negative research grade points, which means that even more people than before need to come across this post, agree with the new identification to make it research grade. And so I'm talking a lot about research grade because it's really, really valuable when utilizing iNaturalist data, because it's essentially showing that, firstly, multiple eyes were on this species and multiple perspectives, and it's more likely to have a correct identification at that point as well. And so now I want to talk about what makes a good observation. Now, good is subjective. You know, it's a describer. This is what I think makes for a good observation when it comes to using iNaturalist data for scientific research. And so firstly, photos. Photos, in my opinion, are the most important thing when it comes to making a post. So some of the ones that we always want to make sure that we have are going to be a top view of the mushroom, an underside photo, a picture of the stem or stipe. Those two words are synonymous. And then a picture of the base of the stipe. And then a cross section where you might be able to observe bruising or staining of a mushroom, or something like ooze, which is also known as milk or latex. So that's just going to be a variable colored liquid that is leaking from the mushroom. And that's oftentimes for certain species a really valuable identification characteristic. And then next, we want to have a photo of the mushroom in its habitat, ideally with the substrate that it's growing out of. So an example of the substrate would be like if you came across a mushroom growing out of a log or out of the ground or a tree or various stuff like that. And then if at all possible, having an age range photo is really important as well, because with that, you're able to see how the mushroom changes with age. Because if you only find a really young mushroom or a really old mushroom, it makes it harder to correctly identify it that way. And then for some species, a veil, if present, is really valuable to have a photo of. So for something like a Amanita muscaria, it has that little skirt-like ring around the stem. And so that would be the veil. And you want to make sure that you're getting that in a photo or your notes just so we know, hey, this was probably an amanita. You probably shouldn't eat this. And then on the side of the notes, I've kind of highlighted the ones that I think are most important. And so that's going to be the color, if there are any sort of color changes over time. So if you've picked the mushroom, you let it sit for a couple hours, you're driving home, and then it's a different color when you get home, that would be important to note when you're making an observation. And then taste. So I'm going to put out a disclaimer. When tasting mushrooms, oftentimes they taste different, which are noted as an identification characteristic. However, this is not me telling you to go be eating mushrooms that you randomly find places. So with this, if you're willing, what you can do is you can take a small bite of the cap, some part of the mushroom, chew it up just a little bit to kind of get the flavor, and then spit it out. And as long as you make sure to spit all of it out, you'll be fine. You know, personally, I don't think I'm going to be doing this with a species that I know is poisonous. And so it's really important to, like, when you're going out looking for mushrooms, be familiar with what your local poisonous species are. However, if you were to do it with a poisonous mushroom, you make sure that you spit it all the way out. There's not going to be as much risk there. But again, this is not me telling you to go eat random mushrooms. So then next we have the odor of the mushroom, which, you know, I often find myself, whenever I find a mushroom, I'm on the ground, I'm smelling the mushroom. It's just something that I do at this point in my life. And oftentimes they are very, very interesting. And so it's another thing to be noting if it has a distinctive smell. In the case of one mushroom, commonly known as the shrimp ruchula, it smells like shrimp. And there are a bunch of other mushrooms in the same genus that look exactly like it, that don't smell like shrimp. And so the scent in that case is a really important thing to be noting. And then the shape of the mushroom, this is going to be kind of the overall stature. And so if there's any weird aspects of it, or if there's any kind of weird texture, well, textures is next. But just if the shape and stature is noteworthy. And that can kind of be determined if you come across a familiar species or an unfamiliar species. You're looking through an ID book. It doesn't quite look like one of the mushrooms in the book, but all the other characteristics are correct. It could just look weird, and it could still be that mushroom. And so it's important to note that the shape was weird. And then texture is going to be, let me just be honest, I don't always include texture. And let me be even more honest with you all right now. Sometimes when I'm making a post, I don't include notes. I don't include anything like that. And I just include the photos, which I think is still important and a valuable observation. And depending on your level of understanding and your level of wanting to be precise, a contribution is a contribution. You're still sharing that you found this mushroom here, which is valuable for documenting biodiversity. Anyways, that was a side tangent. So the texture is going to be, let's say you break open the stem. It's really pithy, or it's really hard, or it's chalk-like. That would be where you would record it as a textural thing. And then next, the habitat. So I think it's always important to, if you're not noting the habitat, to, like, physically noting it, to be observing the habitat, because by observing the habitat, you are making connections between where these mushrooms grow and what they're growing around. And so the habitat in general is going to be What kind of trees are growing around it? What are the plants nearby? What's it growing out of? What's the substrate, which could also be noted as something separate, but still goes within habitat as well. And so it's just, I think it's really important to make those connections between what this mushroom is growing with, why it might be growing with it. Let's say it's a mycorrhizal species. Some mycorrhizal species only grow near certain plants or trees. And so that would be important to note when talking about habitat. And then the mushroom habit, I think this is a weird word to describe it, but it's essentially referring to growth habit. And so this means that essentially, let's say there are a bunch of mushrooms growing in one spot. That would be a gregarious growth habit. There would be a lot of them. And so there are kind of specific words within mycology to describe different habits. And so it's essentially describing how many there are and how they are growing related to each other. And so we have a lot of mushrooms in this area. They're not touching, so they're just solitary mushrooms, but they're growing in a gregarious pattern. And then let's say over here, we have a big cluster of mushrooms. They're all growing fused at the base. That's going to be a completely different habit. And that wouldn't even really be described as gregarious at that point because they're considered essentially one thing being fused at the base as they are. And just in case anyone's curious, when they're fused at the base like that, that is sepitose. That's a sepitose growth habit. I think the names are interesting, so I want to share them with you. Now, the spore print color is always going to be an important ID characteristic. And so regardless of, like, you can't really note that in the field, like when you're just coming across a random mushroom, you can't really determine spore print color. Instead, what you can do is, for example, let's say we have a mushroom that has a cap and stem, has gills underneath. You can remove the cap from the stem, place it in a little tinfoil envelope. So you're completely enclosing the cap, leave it overnight. Most of the time, I make no guarantees. It does not happen all of the time. But most of the time, when you come back to it next morning, around 12 to 24 hours later, it will leave an imprint of the spores released from the gills, and it'll be a certain color. And so when you come across mushrooms that you are not 100% sure they are the same, it's really important to take a spore print from it, because if those two spore print colors are not the same, those are not the same mushrooms in the slightest. And so my example of this is finding bluet mushrooms. Now, bluet mushrooms are these beautiful purple mushrooms. I just personally love any purple mushroom, but bluets in particular are a very tasty edible. They can be made into this sweet kind of syrup, and so it's good with dessert. However, they have a light pink salmony spore print, kind of similar to the color I have for my slides, actually. But the way that they physically looks very, very similar to many species of Cortinarius. Now, Cortinarius mushrooms, we don't eat them. Most of them, if not all of them, are poisonous. And so with me knowing that, hey, I have this purple mushroom, but there are all these other purple Cortinarius, it's super important that I take a spore print. And I've taken a spore print from them, and I've found that I have two different mushrooms. And it's been a very good thing to double check and include, just for personal safety in general, and also just identification. And then I'm going to skip down to the substrate. So we've already kind of touched on substrate, but I want to reiterate really quick. Substrate refers to anything that the mushroom is growing out of. So again, logs, wood chips, pine needles, leaves, just anything that you can note that the mushroom grows from. That's going to be known as the substrate. And then weather, elevation, date. I include these, like, let me go back to my previous slide. I include stuff like that when I'm mainly doing posts for research purposes or for specific projects. So it doesn't necessarily need to be included in every single observation. I think out of those three, if I were to pick only one, I would always note the elevation because some mushrooms are elevation specific. You know, if you are at sea level and you're finding a mushroom and you think it's a mushroom that's growing only at 5,000 feet, it is obviously not that mushroom because it can't grow there. And so it's important when trying to just figure out what you have in regards to elevation. So now I want to go over some example photos. And so this photo is going to be a photo of the mushroom in its habitat. You can see that I tried to kind of include the small plants that were growing nearby, tried to note what it was growing out of, the substrate. And then as for growth habit, we've already kind of gone over that, but this mushroom, one mushroom at first, just wait. And so I would have noted it as growing singly or just as a single mushroom. However, there was a small baby that was growing just underneath. And so technically wasn't single. I would still note it as single because 2 isn't that many. And so then for your photos, you also want to include some sort of measurement. Now, when I'm out in the field, I don't always have a measure tape. If you do, however, that's great. And so you want to measure the horizontal measurements, which is going to be both the stipe width and the cap width. And so you can kind of see, usually when I'm taking these photos, I am seeing how many centimeters it is later when I get home. This is just so I note it in the field. But you can kind of separate how you might measure it when it's horizontal. And then for vertical measurements, you want it to be from the base of the stipe or stem to the top of the cap. And with vertical measurements in particular, like with that baby mushroom on the left, you can kind of see where you might separate the stem from the cap. And that's going to allow you to measure both of those things separately, which is, in my opinion, important to include just for the sake of including it, really. And so next, we want to include a picture of the underside, which is going to be, in this case, gills, but it's really just making sure that you capture the spore-bearing surface. And so that could look actually like a lot of different things. It could look like gills like this, or it could look like little icicle-like teeth hanging from the bottom of the cap, or maybe pores in the case of king boletes. And so it's important to just note what is under there because it could be different regardless of how the top looks. And so in this case, since I was able to find a baby mushroom and a mature one, I wanted to note both gill pictures to kind of see like where the mushroom was fully enclosed and where it broke off from the stem. And so anytime that you can do an age range, it's going to be important for identification. So this photo is of the cross section. And so again, in these types of photos, and when you make a cross section of a mushroom, it's important to be watching to see if any sort of latex or milk is coming out. And so again, that can be white, like milk, or in the case of orange Lactarius species, it can be this red liquid that is just kind of dripping from the gills, dripping from the middle of the stem. And then it can be variable colors, really. There's the indigo milk cap, which has this deep blue indigo latex that it exudes. And so I think it's really cool to just, come across them, do a little slice along either along the full mushroom like this for a cross section, or just to cut into the gills a little bit, and then you can see that latex appear. And then this is also a good opportunity to notice when there's staining or bruising. And so that essentially means that when you cut it open, if it just is randomly a different color later, that's usually going to be some sort of stain. And so back home, we have the aspen bolete, which when you cut it open, it stains dark blue. And so that's a really good characteristic to separate it from any other sort of bolete that might look similar. And then for bruising, that's going to be when you actually put pressure on the mushroom itself. So usually what I like to do for best practice is to just kind of press into the stem a little bit, and then it'll usually change color after a little bit if it does bruise. If it doesn't bruise, stays exactly the same. And then some other important things to write in notes or to at least observe when looking at cross sections includes, but is not limited to, measuring, let me find my mouse, measuring how thick the gills are, like this way across. Because depending on that, again, if any characteristic is abnormal or different, it could literally just mean that you have a different species than what you thought it was. And so it's important to be noting as much as you can to aid in identification. And then when you cut into it like this, sometimes the gills only is what's going to bruise. And so that allows you to have a different perspective on it. And then in the case of this mushroom is actually same genus, same family as our common button mushrooms from the store. It's some sort of agaricus. And where was I going with that? Right. And since I cut it in half like this, it kind of gives a better stature of the mushroom in general. So let me see. We want to give you a comparison. So here we have overall stature of the fruit body, not cut in half. And then I want to toggle back to the previous photo. And then we have this. And so it really just gives you a better idea of how the shape of the cap is, what the shape of the stem is, just overall the shape in general. And so I think it's best practice to always cut at least one mushroom if you have multiple in half to see stature better. And then on the right side, we have an age range photo with my hand for reference. And again, I usually don't just leave my house with a tape measure unless I'm specifically going out to go mushroom hunting or to go look around for stuff. And so my hand, it's always on me. I know that I can always just reference it, and I know how big this mushroom is. Now, we've been talking about how to make an observation, how to make good quality observations, good obviously being subjective. So now I want to shift to talk about how we can kind of use this data for various projects. Now, the example I want to talk about is from a project I did last school year. And so this project revolved around creating a field guide to all of the mushrooms that had ever been observed and recorded on my college campus. And just for reference, that had not been done before. So just a little pat on my back. So I have this question as well, which now I'm going to show you one of those things that you can do with community science data. So for this project, I had to ask an initial question. Now, this initial question was, in general, how many species of macrofungi are found on TESC, is the Evergreen State College? So how many species are found at my college? And by asking this initial question, it kind of brought up these secondary questions. So I wanted to bring up these secondary questions because it helps to better analyze the data that I was working with. And so let's see. The project in and of itself, it came about because during my winter quarter at school last year, I needed to come up with a 4-credit project that was going to be an independent project. I could have worked with other people. I don't always play nice with other people, so I wanted to do it solo. And so for this project, since it was for school, I needed to come up with a specific goal that I could give to my professor. Now, this goal was to create a guidebook to all of the mushrooms found on campus. I was very ambitious when I set that goal. And so I had to kind of take it down a little bit. And so the new goal for winter quarter ended up being to create a guidebook to 10 distinct species from the top 50 most commonly seen. And so this was a lot more accomplishable. Now, to talk about my process, I have this flowchart. So overall, let me again reiterate, there was no sort of species list created at my school. We are a very mycologically advanced school in comparison to a lot of places. We have a very well-developed mycology program, but despite all this, nothing. And so my first step was to collect data. Now, as you can see, that first little square is from iNaturalist. And so I have 4 different sources, first being iNaturalist, the second being the Consortium of Pacific Northwest Herbaria, third being the Mycology Collections Portal, and then EVE is the Evergreen Herbarium, so the herbarium at my school. And so I got all these records, I tallied them all up, and I ended up having 13,077 different records. And let me tell you, this was all just Excel spreadsheets, and they were all formatted in different ways from all of these different sources. And so the next step was to sort it. And so I spent a long amount of time just organizing it into a common format. And then I alphabetized it in fun, confusing ways. Honestly, I would do it differently if I was doing it again. And then I sorted genera into individuals worksheets, and then eventually compiled a full list of all the unique species that had been seen. But they weren't quite all unique because within mycology in general, taxonomy, the act of naming things, putting an identity to something, is shifting. It's shifting all the time and always. And so I had to go through every single species name and see if the name was correct. And so this is just me toggling from tab to tab, seeing if it's correct. If it was correct, great. If it wasn't, I needed to see what the new name was. Oh, it's not a new name, it's actually an old name. And so it was just giving me a big runaround to actually figure out what I had. And so then I tallied up all of the observations for each unique species. And then I made sure that there were no duplicate records. And then I took out all the information I wanted, put it into this big master spreadsheet, which I'll show you now. And so this is kind of the full species list visual that I like to work with. It's essentially all of the information that I felt was relevant for how I was going to use this data. And so in total, I had almost 10,000 unique records. So my original number was over 13,000, took out 3,000 records. Those were just duplicates. They were old names. They were all these random things. And then I ended up with the distilled number of 9,961 unique records. And for the iNaturalist records in general, I wanted to kind of work with the same research grade, non-research grade metric. And so I noted all of that with every single species that I was working with. And so you can kind of see that in the colored column on the far right side. I'm going a little bit over time. I have 5 slides left, I promise. So I'm rapping. And so at the end of all that research, what I ended up with was all this data about what had been seen on campus. And so for a little bit of background, Evergreen itself is located on 1,000 acres of wooded property. And so across all of those 1,000 acres, there were 763 unique species that have been seen, observed, and recorded. And there's a large amount of them, that last bullet point, where there was only one recorded sighting. And so that's a bit tough, because if someone is only like saying, hey, I saw this thing here one time, it makes me doubt if all of those were necessarily accurate, but it's the data that I'm working with. And so it's what I have to work with. And then I want to specifically highlight one species found on campus, this beautiful, beautiful hygrospy species, Hygrospy flavifolia. And I want to highlight this specifically because it is endangered. And the only reason that we knew that it was on campus at all is because of iNaturalist. So it's been observed 2 times so far, once in 2021, once in 2022. And across the board, we really just would not know that it was there without people going out, looking at their local mushrooms, trying to figure out what they are. And so this kind of gives us a lens into how we can use community science as a way to further fungal conservation. So I also have some example photos from my book. I'm just gonna flip through them pretty quickly, just so you can see how I was doing things. But I mainly want to get to talking about conservation. So this was the cover. This is a map at Evergreen. And so all of that colored portion is the actual property that the school has. And so that's the 1,000 acres that I was previously referring to. And then just going to briefly tell you how to understand it. So I split up each species page with 4 main characteristics: identification features, ecology and habitat, comments, and then morphology references. So the key ID features: anything you'd ever want to know about what this mushroom looks like. Ecology habitat: anything you would ever want to know about where this mushroom is growing, how they grow, what they're growing with, virtually anything like that. Comments: my soapbox. For the comment section, I'm really just talking about the species through my personal lens and how I view them. And so that was honestly my favorite portion to write. And then the morphology references, all of those acronyms, I'll toggle back to the previous page, are from the morphology references over here. And so it's essentially me listing all of the guides that I used to create the identification characteristics, because I don't want to be putting out information about a species, especially if I've not personally seen it. And so I'm using references. And so that's what that section is. And so we have Fomitopsis mountziae and Caprinus comatus, two common species found on campus. And then we have Aluria aurantia and Ari scalpum, two less commonly found species, but I think they're rather cool. And so I wanted to share them with you today. I am low on time. And so I don't think I'm really going to get into my big soapbox about mushroom picking etiquette, but feel free to take a picture of this if you'd like. Also, feel free to ask me for the slides if you'd like later. I'd love to share them. But these are just some separate areas when picking mushrooms that I think it's important to keep in mind. What I want to highlight are the best practices. So avoid picking near roads and lines of sight. You know, these are beautiful things most of the time. And so it's important to allow other people to also enjoy them. And then considering contamination risk is important when foraging for edible mushrooms. You want to be mindful where you're picking because mushrooms are really, really great at picking up things that we would not want in our body. So that's going to be chemicals, heavy metals, various things like that. And so you want to avoid sketchy areas. Don't pick near roadsides, don't pick near industrial sites, things like that. You know, it's best to use your own judgment when it comes to it. And then leave no trace when you're out there. Pick up micro trash, pack in what you pack out. Opposite, pack out what you pack in. So yeah, these are some of the best practices. And then these are my favorite guides when it comes to identifying mushrooms in this area. All of them are super, super important. In particular, I want to highlight that the Pacific Northwest Key Council is a free guide. It is in the style of a dichotomous key, and it's online. You can just look it up. And so it's essentially the type of guide where you'll have two options when it comes to a mushroom. So let's say the mushroom has a cap. Does it have teeth underneath? Does it have gills underneath? You pick which option best suits the mushroom that you have, and you do a similar series of picking an option until you get to your species. And so it's super useful when trying to separate it from a different species. So now, the last section, I want to talk about why fungi are hard to conserve and what we can do better when it comes to conservation. So some of the challenges that we face revolve around taxonomy, distribution, and ecology. We're just really uncertain about these three main things. And so taxonomy, again, the naming of things, if we're always renaming things, we don't really know what anything is. And so if we don't really know what anything is, we don't really know their distribution. If we don't really know what their distribution is, we don't really know what their ecology is. And so it's kind of this trickle waterfall that happens when things are in flux as they currently are. And then also to successfully conserve fungi, you need to also conserve associated partner organisms. And so if an animal or a plant is the host of a specific fungi, guess what? We need to conserve those too if we want to conserve that species. And then any sort of mycorrhizal association, if we lose the mycorrhizal host, or if we lose the fungal, like mycorrhizal partner, it's a good chance that the other partner will be struggling to survive after that point. And then this goes hand in hand with conserving habitats as well, because if we're losing full habitats at one time, then it's really easy for fungi to just disappear before we even knew that they existed. So I quickly want to highlight some understudied fungal habitats. these are all habitats that we just don't know much about. And until we get people who are either super excited about these or just willing to explore, willing to learn, we probably won't really have too much new information about them anytime soon. Which is, in my opinion, when community science comes hand in hand with conservation. And so, in general, at current with fungal conservation, the IUCN Red List is the International Union for Conservation of Nature's Red List of Threatened Species. Long name, long title. And it's essentially the most comprehensive list of any sort of endangered species to date. And so it was established in 1964, and it's assessed over 169,420 different species. And so you can see from this image that from that number, 47,000 species are currently endangered or threatened with extinction, really. And so that is 28% of all the assessed species in total. So 1,300 species of fungi have been assessed, which is 0.76% of all assessed species. A very small portion of that. And then out of that 1,300, 48 of those species are critically endangered. So we are at threat of losing those species to extinction at current. And so this is just kind of the levels of assessment that the IUCN Red List operates with. It's virtually just a range. Every species that is ever assessed will get assigned one of these. And reassessments are also happening pretty often, honestly. They try to reassess a good amount of species each year. And so oftentimes you might see a species that is considered vulnerable, and the next time it's assessed, it could be either near threatened or it could be endangered, critically endangered. And so the reassessments are really important to evaluate what is happening with the species. And if they are either further threatened or having a come up, you know? And so now I just want to highlight some of the other programs that are around that are working on fungal conservation. And so we have the Washington Natural Heritage Program. That's going to be more of a local program within Washington State itself. It works with this 4-step system, and its main goal is to create a registry of all natural habitats with all of their associated organisms. And so again, this is Washington State specific. So it's just going to be a big catalog of what's where, what is there, you know, where things are and what is there. And then next we have NatureServe, which collects, analyzes, and delivers biodiversity knowledge to inform conservation action. So they also have a pretty good program dedicated towards conserving fungi. It's like in and of themselves. And then the Global Fungal Red List Initiative is a sub-program under the IUCN Red List, and it exists specifically to track fungi and all their conservation needs. And then the Fungal Diversity Survey is the main one of these that works with the most community science efforts. And so they work to protect fungal diversity through conservation, education, and community engagement. And so it's a super powerful one in the mycological world at current. It's a nonprofit, and they're just super cool people there. I've met a couple of them, and I really appreciate what they're doing for fungal conservation. And that's it. No more slides. Thank you. It's 7:30, but if anyone has questions, feel free to talk to me to the side. But I know I want to respect everyone's time, and this is our cutoff point. So again, thank you very much for coming.